Good afternoon, everybody, and welcome to this ICTS Distinguished Lecture by Jennifer Chayes. I'll briefly introduce her. She's Distinguished Scientist, Managing Director and Co-founder of Microsoft Research New England and Microsoft Research New York City. And before joining Microsoft in 1997, she was for many years professor of mathematics at the University of California at Los Angeles. So she has authored 125 academic papers and the inventor of 30 patents. Her research areas include phase transitions in discrete mathematics and computer science, structural and dynamical properties of self-engineered networks, graph theory, graph algorithms, algorithmic game theory, and computational biology. It's an amazing sweep, and it is an extraordinarily interdisciplinary scientist, and this is, I think, what attracted me to really invite her here, because I had heard her talk at the Institute for Advanced Studies in Princeton. So she holds a BA in biology and physics from Wesleyan College, where she graduated first in her class, and a PhD in mathematical physics from Princeton, postdoctoral work in mathematics and physics departments at Harvard and Cornell, recipient of the NSF Postdoctoral Fellowship, Sloan Fellowship, UCLA Distinguished Teaching Award, and the ABI Women of Leadership Vision Award. She has twice been a member of the Institute for Advanced Studies at Princeton. She's a fellow of the American Association for the Advancement of Science, Fields Institute, the Association of Computing Machinery, ACM, and the American Mathematical Society, and an elected member of the American Academy of Arts and Sciences. So let me tell you that she's on a very, very short visit to Microsoft Bangalore. And when I mentioned that she's going to visit us to Ravi Kannan, his jaws dropped and said, oh my God, so much time away from there. But thank you indeed for coming over to give this, I'm sure, fascinating talk. Thank you, Jennifer.
Can you hear me? Yes? Okay, good. So thank you so much. Seems like an absolutely lovely place. Can't believe that this is in the middle of Bangalore. Bangalore always seemed so dense to me. And you come here and it's this wide open place with all these beautiful trees and everything. And I know that ICTS will not be here forever, but it will also be in a beautiful campus in Bangalore a few years from now, on its own campus. But I'm very happy to visit ICTS here at the IAS. Okay, so age of networks. So my motivation is that everywhere we look, we see networks. We see the internet, the World Wide Web, we see social networks, we see biological networks, we see resource allocation networks, mathematical networks. So I'm first going to tell you about the observed networks. So what are the networks that I'm going to try to model for you? Then I will tell you about 5 different classes of problems on these networks. And then I will focus on one specific problem or class of problems. Okay, so the observed networks. So we're going to be looking at technological networks, at social networks, at economic networks, and at biological networks. So I'm a mathematician, so I model these networks as graphs, so they have vertices and edges. An example of this would be the AS Internet. So here the vertices are autonomous systems like Microsoft.com is an autonomous system. AOL.com is an autonomous system. They can be walled off. The edges are the connections from one autonomous system to the the other. These are bidirectional. The World Wide Web is a directed graph. The stationary web pages, not the ones that are generated, when you go onto a page, but the stationary ones are the vertices of that. And the hyperlinks take you from one page to another, but they only go in one direction. The cloud data center networks, there's a huge network of data centers out there. This is a really interesting topic for study. In the US, somewhere from 3 to 5% of the power consumption in the US is in the cloud right now. When you do a web search, you think it doesn't use any power. Isn't it amazing? It finds these answers for you just out of thin air. But of course, there are all these calculations going on. And so it's very important that we understand these networks, these data center networks, and how to do the computations more efficiently on those networks, because the amount of power that we are using in the cloud is doubling every 5 years. Okay, then there are social networks. There are social networks that have been studied for years, offline social networks for the spread of disease. We still study these. Whenever we have some kind of an epidemic, we study these. There are online social networks, Facebook, LinkedIn. There are mobile phone networks that people have studied, instant messaging networks, microblogging networks. Each of these has a somewhat different structure, and due to that structure, there are different kinds of processes that you can observe on these networks. And there are really nice problems in which you model these, and then you can also gather data and see how the models perform. Okay, there are economic networks. Now, a very interesting economic network happens on the autonomous system internet. So there are 3 levels of the autonomous system internet. And if you are on the bottom level, the 3rd level, then to have your traffic carried through the 2nd level or through the 1st level, you need to pay. And if you're on the 2nd level, you need to pay to have your traffic carried on the 1st level. So what happens is that you get all kinds of strange routing based on the incentives, the economic incentives. And so if a priori you were trying to figure out how some traffic would be routed and you didn't take those into account, you would get something very different from what is actually observed. There are a lot of people who study the economics of this network. Then there are just bipartite graphs of buyers and sellers and market networks. So the ad network that we have on Bing or on Google, of course, there are ads, and then there are eyeballs or customers who come in, and those have to be paired up in various ways. And so all of those are economic networks. There are implicit economic networks that underlie those. Okay, then there are biological networks. So there's the phylogenetic trees, which have been studied for many, many years. We look at the species that are present today, and from those we try to infer how they must have evolved. Okay? So that's actually a combinatorial optimization problem. There are gene regulatory networks. Okay, so there's DNA is transcribed to RNA is transcribed to proteins, and then the proteins go and sit on the DNA, and we get an interactome. And there are real neural networks, and this is becoming a huge area now. The amount of data that we are getting from real neural networks will dwarf the genome data. Okay, so what are the mathematical and the classes of mathematical and algorithmic problems on these networks? So what I'm going to do first is just talk about how we model these networks. Then I'll talk about sampling from the networks. processes on networks, so various kinds of processes that we're interested in on the networks, algorithms on the network, so algorithms that one can design that occur on these networks, and finally, network reconstruction algorithms. Okay, so modeling. So if I'm going to model something, I want to know what properties I'm trying to model. So I have to tell you what some of the observations are that I would like to make sure are in my model. So, okay, one of the things that people see in many of these modern networks is that they have a small diameter. So people talk about 6 degrees of separation. So Milgram did experiments in the 1950s where he gave a postcard with the occupation of someone to someone in a different part of the US. So for example, in the Midwest, he would say, this is a school teacher in Massachusetts. Send this, or take this postcard, send this postcard to the person you think is on the shortest path that you can find from you to that person, okay? And so this is a local algorithm because I can't see the whole network. I only know the people that I'm connected to. But what you might try to do is you might try to send it to somebody you know who's really well connected and have them send it down to other people. And he found that there were basically 6 degrees of separation. Now, in fact, in 1929, a Hungarian author had already hypothesized in a short story, or suggested in a short story, that everybody in the world was separated by no more than 6 degrees from everybody else. And then a couple of years ago, there were some experiments on Facebook which showed that there were about 4.5 degrees of separation on Facebook. Okay, a second thing that we want to account for is the power law degree distribution. So if I ask, what is the probability that I have k neighbors? Turns out that that falls off like a power of k, which is pretty slow relative to some distributions that we know well. The simplest things that you can think of have exponential fall off, so much, much faster. Okay, then aging of vertices. So on the AS internet and the World Wide Web, older vertices tend to be more highly connected. And in fact, this is how web spammers work. They buy up old vertices on the World Wide Web, old domain names that people haven't paid for. So if you don't pay, you lose the domain name. And they try to put a lot of edges from those old, well-connected domains to your web page so that there's a better chance that a search engine will land up on your web page. If you don't understand that yet, whoops. Let me tell you very quickly how search engines work. Probably most of you know this. The early search engines used content and language to find the most relevant web pages. So search engines like Alta Vista used that. Then Yahoo and Google came along and Bing came along, and they used the structure of the web graph, so graph theory and algorithms, to find the most relevant web pages. PageRank is a very simple algorithm which just says do a random walk on the web graph, so follow the hyperlinks. And you might get stuck if you do that. You might land up on a page that has no outlinks. So every 7th step or so, or with probability 1/7, you should jump to a uniformly random web page. You keep on doing this for a long time, and you eventually land up at a stationary distribution. Your rank in that stationary distribution is your page rank. So if you have a lot of inlinks, you have a very high page rank. If you have very few inlinks, you have a low page rank. that's to first approximation the result of this stationary distribution, but it's more complicated than that. And so what you can do is so what the web spammers do is they buy up these old domain names and they create extra inlinks, which will artificially elevate your page rank. And so there are algorithms that you can come up with, and this is something that I worked on a few years ago with John Hopcroft and Shanghua Tang and some other people, in which you detect anomalous structures as you're doing the random walk, and you actually just downweight those slightly. And when you do that, then you don't have to manually go in and pull out this web spam. Okay, so modeling networks. Okay, well, the first model was the Barabashi-Albert model. Really, really simple model. At each time step, a new vertex comes in, and it attaches to m old vertices. And the probability, so this is like a rich gets richer model, the probability that a new vertex attaches to an old vertex is proportional to how many neighbors that old vertex has, okay? So this is like, you walk into a room and you go and you join the conversation that has the largest number of people in it already, because you figure, oh, that's probably an interesting conversation. So then you get a distribution with long tails. Okay, and the first rigorous work on this was a year later. Okay, so there are other types of models. So that was a simple probabilistic model. You can have more complicated probabilistic models. So there could be a preferential attachment model with fitness. So what Bianconi and Barabashi said is not all web pages are born with the same fitness. Okay, there are some web pages that a lot more people are going to want to attach to than others. So let's look at a distribution of fitnesses. And each time a page is born, it's also going to be born with a fitness that scales how attractive that web page is. And so we did a rigorous analysis of that a number of years later, and it was really interesting to the physicists here. It had a Bose-Einstein condensation. So what happened was the same integral that shows up in the Bose-Einstein condensation where, a positive fraction of everything pours into the ground state, what you get exactly that same integral appearing. And what it means in this case is that a positive fraction of every future page that's born is going to attach to some web page. So there's some web page that's going to get a huge amount, a positive fraction of all future web pages that will attach to it. And you get that in a certain regime of distributions, and in another regime, you get something that looks like the ordinary preferential attachment model, and you have something that looks like the Bose-Einstein condensation as you go through that change. Okay, then, so there are probabilistic models. There are also competition models. So you can say each time a vertex comes in, instead of just attaching randomly, what I'm going to do is I'm going to try to optimize some function, okay? And so you can you have some function which has different parameters in it, and we were able to show that when you have models of this form, you can actually fit a lot of data that you couldn't fit otherwise, like actually for the structure of the AS internet, these models gave us the best fit. So that's when a site comes in once and for all, it optimizes a given function and does the attachment based on that optimization. Now, if you do a fully game theoretic model, then you don't do this once and for all. You come in, you try to optimize something, and then the other sites come in and they optimize, and then you re-optimize on the basis of what the other sites have done, and you keep doing this again and again until you get to an equilibrium, a Nash equilibrium. Okay, so it's not a one-shot optimization. It's you're optimizing and re-optimizing till you get to some kind of an equilibrium. So those are fully game theoretic models. Those are much more complex, and there's been much less work on those. Okay, so we have different kinds of models. We have probabilistic, we have competition models. So now one of the questions that one really cares about is, how do you sample from a large network? Okay, if I wanted to do something really simple, like compute PageRank, which is just finding the stationary distribution of a random walk. If I wanted to do that on, on the web, which has over 1 trillion stationary web pages, that would be a really difficult thing to do. So what I would like to do is I would like to sample in some way. And so when you start to think about sampling, how would you do sampling? Well, in order to deal with this, we developed a theory of graph limits in testing, and I was just talking to some of you earlier about this theory of graph limits, which was in fact motivated by thinking of what would a thermodynamic limit of a graph look like and what would be the properties of that limit. And once you have a limit, then you can sample from it in a nice way because you have the stationary properties of this limit. And so over a number of years, we developed a theory of this, and the limits of these graphs turn out to be just measures on the unit square. And then more recently, we were able to get graph limits for sparse graphs with power law tails, and those turn out to be unbounded functions on the unit square. And once you've got these nice functions, then you can sample very easily from them, from the distribution that acts like this graph in the limit. Okay, so that's another class of problems, is sampling from large networks, and there are now hundreds of papers on these graph limits. Okay, so another class of problems is processes on networks. The networks themselves are often generated via a random process. The preferential attachment graph is a random process. But on top of this thing, which is randomly generated, you can have a random process, like the flow of information. And John Kleinberg has done a lot of work on the flow of information on networks. Okay, you can also look at the spread of epidemics on networks. You can have a virus or a worm that transmits from one site to another, and you ask, how does this random process on this random network? So you pick out a representative sample of your random network and then run a random process on it and ask how it behaves with high probability with respect to the underlying network. Then there are also questions of viral marketing. Okay, so if I wanted, so this is kind of an inverse to the spread of epidemics. If I wanted something to spread very quickly, where would I put it? Okay, where on my graph would I put it? And so then if you have a social network and you want to get adoption of something, this would tell you where you should put that to have maximum spread. So they're mathematically similar but different problems. Okay, then there are algorithms on networks. So when I have a network, I can do an algorithm on it. We already learned one algorithm, which is the PageRank algorithm. Okay. There are, in fact, more sophisticated ranking algorithms. So in the past few years, a lot of people have been looking at questions of, can we not get a perfect answer, but can we get an okay answer in very, very fast time? Time as a function of the size of the network, okay? So we, for example, have a sublinear time PageRank algorithm, sublinear in the size of the network. Okay, then there are all kinds of clustering algorithms or collaborative filtering algorithms on bipartite graphs. So Amazon uses collaborative filtering all the time. You buy this and that, and so it thinks that you're also, it does a clustering, and it looks at people versus products, and it says, oh, you're probably also going to like this, okay? Or Netflix, it's a matrix completion problem, right? You have movies and you have people, and it's a sparse matrix because you haven't seen that many movies, but it gives you a completion of that matrix. So there's beautiful mathematics there and computer science, but also it has commercial applications. Okay, then there are algorithms for multicasting. So we've done some algorithms. I hear this is being multicasted. So there are algorithms for multicasting in a really efficient way, which can actually be turned around, and I will be turning those algorithms around to find out something about the structure of networks later in the talk. And there are also algorithms for web hosting. So Tom Layton and his student just had a stock paper or a Fox paper saying, okay, if I wanted to mirror content in different parts of different parts of the internet, where would I put it so that you wouldn't have everybody trying to go to the same site at the same time and bringing the site down? And this is just an algorithm for how you would do that mirroring, okay? So you see that like all of these algorithms have led to big companies, okay? Then there are other kinds of algorithms. There are fast algorithms for identifying influential sites. So this is viral marketing, okay? How do you identify the influential sites? And there are also algorithms for recommendation systems. So if you have some kind of a social network or an online trust network, how do you generate recommendations based on that network? Okay, so all of these, they're nice algorithms from a mathematical or a theoretical computer science point of view, but also they form the basis, and then there are thousands of tweaks, but they form the basis for different kinds of businesses. Okay, then there are network reconstruction algorithms. So we talked about phylogenetic network reconstruction, okay, looking at the leaves of a tree, and from that, trying to conclude how those must have evolved, where the splits happened. Okay, there's gene regulatory network reconstruction. So I look at what protein is present with what other protein, and from that, try to infer the gene network that produced it. And there are also reconstruction of learning processes and networks of synapses. So, they can do this on a little worm that has about 300 neurons. They've mapped out all those neurons, and then they put this little worm in different kinds of situations and see how it reacts. And from that, they have simple models like perceptrons and then more sophisticated models, and they try to infer what the learning process was on those 300 neurons. Okay, so now I'm going to focus for the rest of the talk on these gene regulatory networks, okay, and reconstruction of gene regulatory networks. So, that's interesting. I don't know how that happened. Okay, so the standard dogma here is that DNA goes to RNA, goes to proteins. And so what happens then is that the proteins that are transcribed from the RNA go and they sit on the DNA, and they affect the transcription of the DNA into RNA. So they can either speed it up or slow it down. So we get an interactome, a protein interactome. Okay, so problems with gene regulatory networks are the source of many diseases. So how can I infer from partial network data, how can I infer the network structure? And also, can I identify particular nodes in the network which are responsible for the dysregulation in certain diseases and in certain individuals. And are one or more of these nodes in combination viable drug targets? So those are some of the questions that we'll be trying to answer. Okay, so there are many different kinds of data. There's microarray data, there's mass spectrometry, there's all kinds of data. From that, we use computational models, and that suggests points of intervention in the network. And one of the big problems is that different classes of data often suggest different points of intervention. It doesn't mean that any one of them is wrong, it just means that they're suggesting different points of intervention, all of which may be legitimate points of intervention in the network. Okay, so let's look at gene expression data. So microarrays tell us which gene is expressed in the presence of which other gene under a particular set of circumstances. Okay, so this gene is getting transcribed while that gene is getting transcribed. And from the differential expression of a given gene, so the expression relative to the normal background, I can infer a node weight of a corresponding protein. So I say, you know, if something is being way overexpressed or way underexpressed, it's interesting. And so I'm going to flag the corresponding node. Now, for the edge weights between two different proteins, what I'm going to use is how often in everything that has been measured about that kind of organism, how often does it seem that these two proteins are interacting directly with each other? This is very dirty data because sometimes we think they're interacting directly and they're actually interacting indirectly. Sometimes we actually see direct interactions, like one protein will be complexing with another. Sometimes we only see something downstream of that. Okay, so the edge weights are going to be constant for the organism, whereas the node weights are going to differ in each realization. Okay, and how do we determine the network most likely to have produced that data? So, okay, we're going to use a model called, or a problem called the Steiner tree problem. So you have a graph, you have costs on every edge, positive costs, and then I have a preferred set of nodes. So out of all the nodes, out of all the 2029 nodes, here are 432 nodes that I want to have in my solution. Okay, the others can be in there or not. And what I want to do is I want to find a tree which contains all the special nodes, the 432 nodes, and which minimizes the cost of all the edges in between all the nodes used. So it'll be those 432 plus whatever other ones that I've used to construct a tree. In general, the minimizing tree will contain other nodes in addition to the terminals. And these additional nodes are called Steiner nodes. So 432 have to be in there. But let's say the minimizing tree I come up with has 775 nodes. Okay? So 775 minus 432, those extra nodes are called Steiner nodes. And there are computational issues here. This turns out to be, this is a global problem because I need a tree overall. So you would think that you wouldn't be able to compute it locally, but we found a new representation of the model where you could, by replacing scalars with vectors in a certain way, you could turn it into something that had a local algorithm. And you could do it in a distributed fashion, and then we were able to get much faster computation. Okay, so what is going on here? Oh, here we go. This is interesting. Let's try this. Do we have a technical person here? Oh, and there it goes. I don't know what's going on, but it seems to be fine now. Okay, so biological problem. So now I'm going to do something a little bit more sophisticated than the plain old Steiner tree problem. I'm going to do the prize collecting Steiner tree problem. So instead of having 432 privileged nodes, and then everything else had no privilege whatsoever, I'm going to give nodes weights. And the higher the weight, the more I'm going to have a tendency to include that node in my solution. Okay? So what I do here is that I have prizes, and because I'm trying to minimize this, and this lambda is positive, anything with a prize, with a big prize, I'm going to want to keep in the solution. And as lambda goes to infinity, this is just the good old Steiner tree problem, because then I need to include everything with a positive prize. Okay? So these 432 of them have positive prizes, but those prizes might have different amounts, and I really want to keep the ones with big prizes. Okay, so you know what happened? The animation on this, for some reason, got screwed up. So for reasons that I do not know, but okay. So what I'm going to do to map it to biological data is I'm going to find the tree which minimizes this. Now, what are the CIJs going to be? Remember, the CIJs were supposed to tell me how likely it was that protein I interacted with protein J ever in this certain organism, okay? So it's going to be, so CIJ is going to be minus the probability that a connection exists directly from protein I to protein J. And this is going to be something that is compiled from all the experiments ever done on that organism. So these CIJs are very dirty data, okay? And then the prizes are going to be particular to the experimental realization. So I'm going to run one experiment under this set of conditions, and I'm going to have this protein and that protein being expressed different amounts. Then I'm going to run it under another set of conditions, and I'm going to find different levels. And the more a protein differs from its background value, I don't care if it's above or below, the more it differs from its background value, the bigger the prize I'm going to give it. Because what I'm thinking is, if it's differing a lot from its background value, it may be involved in whatever interaction I'm looking at, okay? So, wow, this is really weird. So all of the animation is screwed up on this, but okay. So in the standard Steiner tree problem, the nodes which are included in the minimizing solution, but which are not terminals, okay, which are not in the set S, are called Steiner nodes, okay? In the prize collecting Steiner tree problem, what I'm going to say is that nodes which don't have a big prize, so, and I have to tell you what my definition of big is, nodes that don't have a big prize, but which land up in the solution are Steiner nodes. And so this guy here is a Steiner node because, let's look, I'm supposed to minimize the sum of the CIJs, so I don't want these heavy things, okay, because I want to minimize the sum of those. So as I'm coming up with a solution, what I do is I don't take any of these really fat edges, because that costs me. But it's minus the prizes, so I want these big nodes here. So I'm going to want to try to get all the big nodes. Now, what is this? This is a little node, so by itself I wouldn't want it, but what it does is it brings these two big nodes into the tree, okay? So in this case, I say that that is a Steiner node. It's a gene which is not differentially expressed very much, but which nevertheless seems to be participating in the network in some way. So this is identification of proteins which are not previously known to participate in the pathway. Okay, now we'll see how the animation works here. Okay, strange. Good. So the first example, what we did was we looked at some yeast data, okay? And for this, we could just, go and find existing data on the yeast pheromone pathway. Yeast is very, very widely studied. So yeast, there are about 5,000 known proteins, okay? And there are about 15,000 known protein-protein interactions. So that determines the CIJs for me, just from the organism, okay? Then there were 56 different experimental realizations where we found different genes expressed in the presence of other genes. For each one of those 56, we got a set of prizes, okay? We have the CIJs, we have a set of prizes, we find a tree. We have the same CIJs, a different set of prizes, we find a second tree, and we go on. We find 56 trees. We made them bounded diameter for various reasons. And we merged them to get one network, okay? The network is no longer a tree because it's merging 56 trees. So let's see how it looks without the animation working. Okay. So here was the pathway that we identified. There were two types of proteins on the pathway. There were proteins that we had expected to find because they had big prizes. They were differentially expressed. Okay, so we knew those were going to show up. But then there were proteins that were not differentially expressed but seemed to bridge different parts of the network, like that little node that I surrounded with a red circle, which brought in some other high prize nodes. So this one is one of those in this network. And so the question is, are these Steiner proteins important in the pheromone response pathway? Okay, so, wow, this is weird again. Okay, so we did an experiment to knock out the gene corresponding to that particular protein that we identified. And it turned out that the pheromone response pathway failed. So this was experimental proof of the importance of the Steiner node, okay? Now let's go from yeast to mammals. Well, the problem is with mammals, okay, so yeast is fine and, it's nice if we get some results there, but mammals are actually much more interesting. But they have problems. They have incomplete interactome data. They have 10 times as many transcription factors. They have huge regions in between the genes, so you need much faster algorithms. Okay, so the second example is brain cancer, glioblastoma. It's the most deadly brain cancer. It's the kind of the common human cancer with the worst outcome if you have surgery, which means going into your brain and taking something out, which typically has very dire consequences, and chemo and radiation, you live about a year. And it's much more common in men than in women. Okay, so can we find the glioblastoma pathway using the prize-collecting Steiner tree? Okay, well, how do you choose it turns out you have to choose a route for the prize-collecting Steiner tree. And what do you want to choose as a route? You want to choose some protein which you think should be very important in a signaling pathway. So a good thing to choose is a membrane protein because typically signaling comes through a membrane protein. So EGFR, if you know someone who has cancer, you may hear very often, oh, is this EGFR sensitive cancer? It's a variant which is a very common variant in human cancers, a mutation of EGFR, and it happens to be present in 60% of glioblastomas. So we use that as the root of our Steiner tree. And this was the resulting pathway that we got, okay? It's a pretty complicated looking pathway. What happens is that if you are a biologist and you look at this pathway, you see that there are many parts of it which make a lot of sense to you. Okay, there are things that are involved in DNA damage repair and nuclear transport and things like that, and you say, oh yes, if there's a problem with that, you know, it's very likely that you're going to have a serious dysfunction. Okay, so in this picture, it's a little different than in the other pictures. The size corresponds, the size I didn't know in advance, so it's not the prize. The prize is indicated by the color. So EGFR, I put a huge prize because that was my root. Anything that's pretty red had a big prize. Anything that's white had no prize. And then the size has to do with how central it was, certain measure of centrality, how central it was in the resulting network. Okay, so you might ask, how about things which are very central but had very little prize on them? Okay, like this guy here, okay? So, turns out that the top 5 nodes ranked by betweenness centrality are those. The first one was well known to be active and it had a relatively large prize. ESR1, which was that one that I identified for you, had no prize. It was not previously known to be important for glioblastoma. And it's the estrogen receptor. So this was the first pathway link between glioblastoma and gender, okay? So it was 4 times as common in men as in women, and there had been no link, no pathway link of gender and glioblastoma. And so then we took estradiol, which is basically estrogen, and EGFR inhibitor, and not we, but our collaborators, and put it in culture with glioblastoma cells, and the glioblastoma cells were killed off much faster than with the EGFR inhibitor by itself. So, you know, I don't know, can you get estrogen into the brain? Who knows? But it's a possible drug therapy. Okay. So, sorry about that. This is doing weird things here. Okay. So we also looked at multiple signaling pathways. So we know that there are a lot of parallel pathways that go on. They can be triggered by different membrane proteins, for example. So then instead of using a prize-collecting Steiner tree, we used a forest. So we had many different trees. And we found a solution of this. You could have added a fugacity for the number of trees, or you could fix the number of trees. And so we implemented it by putting in an artificial node. And this might do really weird things with the animation messed up, but we'll see. Okay, so here is the artificial node. And then when you get rid of the artificial node, what you find is you find these separate trees that are parallel pathways, some of which are Steiner nodes, so the hidden Steiner proteins, okay? And so we did this first with the yeast pheromone network, and we found these different pathways that made a lot of sense to people. And then we did it with glioblastoma, so this was related to the old pathway we found, and then we found some distinct parallel pathways to it. Okay, and then the final thing we did was we tried to come up with patient-specific networks, and we looked at the breast cancer data from the Cancer Genome Atlas, which is a big collaborative effort by 20 or 30 different institutions that are all gathering data. And so what we did was we learned the networks of individual breast cancer patients. So for each breast cancer patient, we learned its individual forest, and we had individual Steiner nodes. And then we took these forests and we clustered them, because we know that, you know, there are different breast cancer clusters, right? There are some breast cancers are susceptible to certain drugs and others are not. There are all these things which we categorize as breast cancer that are probably different diseases on a molecular level. The same thing is true of lung cancer or prostate cancer. There are all these different things that we just lump as the same disease, but on the pathway level are probably very different. And you really want to discover that because they respond to different drugs. And if you can't figure out that these are different diseases, you don't know which drugs you should be giving. So we said, okay, once we get the forests, let's cluster them into similar forests. Now, in the similar forest, for each forest, we find some Steiner nodes, okay? We find some proteins that we didn't think were going to have anything to do with it, which actually do. So then what we do is that we add prizes. So we say, those we didn't know in advance were important, but we've now found out from some other class of breast cancers that those are important there. So now let's add weights, let's add prizes to everyone, to all of these clusters for those nodes, and let's iterate, okay? So we are enhancing the prizes for the nodes that show up from the other kinds of breast cancer. And we iterate this until we settle down on solutions. And what we found was then highly patient-specific networks, which have inputs from the networks of other patients. And interestingly, we found a subclass whose Steiner nodes implied that they could be treatable by drugs that were already developed for a particular kind of gastrointestinal tumor. So this is really useful because if there's a drug that's already been developed, it's already gone through, stage 1, 2, 3 tests by the FDA, by the Food and Drug Administration. So you already have all the tests about whether it's toxic or not. And the only thing you have to worry about is it effective against that kind of cancer? So what we found was that we found a particular part of the network in one strain of breast cancer that looked like this part of the network in this gastrointestinal tumor for which there was a drug. So now we're trying to get somebody to try this drug on that strain of breast cancer patients. Okay, so everywhere we look, we see large-scale networks. The modeling and the analysis of these networks use approaches from many different kinds of mathematics and computer science. The results give us new theories, new theorems, new experimental predictions. As we saw when we looked at those algorithms, which each had given rise to, major businesses that are in the Fortune 500, we can get new business models and possibly new personalized drug therapies. Okay. Sure. Over there? Yeah?
One minute. Let others everywhere else hear you.
So the example you showed, are these directed networks, and how do you deal with uncertainty in the networks, which is inevitable?
Okay, so are these directed networks? They are not, in biology, of course, they are directed networks, but the data is so dirty that we haven't felt sufficiently confident in them to use the directions that we have. We are just starting to get some data that we think is good enough, and we've now done variants of the Steiner tree algorithms that allow us to do that. So we're just starting to do that now, but the data we've had so far we felt just wasn't good enough to infer the directions in the networks. We believe that when we are able to do that, it's of course going to be a lot more accurate.
So in the slide where you introduced the Steiner tree problem, it said that in general, the minimizing tree contains some Steiner nodes.
Contains some
contains Steiner nodes,
Yes.
other
Yes.
nodes. So are there conditions where it does not contain any other vertices? I mean, anything is known about that?
Okay, so the question is, are there cases in which there are no Steiner nodes? Well, I can give you cases where you must have it because these, I can give you like 3 nodes that just can't be connected directly. And in general, the odds that we would have a tree that would be able to connect these particular nodes that have all been very underexpressed or very overexpressed are very, very small, just because there are a lot of nodes and but they can be in very different parts of the pathway. So we haven't run into that, and I don't think that would happen very often because typically it's a, relatively small percentage of the nodes that have prizes, that have large prizes.
So typically these Steiner nodes have higher betweenness centrality. And then if in a network, how do we found that which one is the Steiner node if there are many nodes which have higher betweenness centrality?
So there's no guarantee that, that any particular node with high in-betweenness centrality, I mean, it'll be a Steiner node, but that it will be important in the network. So when we do experiments, or when the biologists who collaborate with us do experiments, there's no guarantee that the first thing that we identify is actually going to be biologically interesting, or that we're going to be able to identify the way in which it might be biologically interesting. It's just those are the things which are most likely to be biologically interesting. So we go from, the things which have the highest in-betweenness centrality with the lowest prize, and we just, we continue down the line until we find something that is interesting. And we've been lucky in that the first things we've identified have turned out to be interesting, but there's no guarantee of that.
Why should the output of your algorithm always be a tree and not probably a graph with some connectivity
The
properties?
Steiner tree, why would I include an extra Cij making a cycle? Because I'm trying to minimize the sum of theCijs. So I would never make a cycle because I'll just be adding an extra positive term for that Cij. But then when I take many of them and I superimpose them, then that's of course not a tree. But if I'm trying to minimize the sum of theCijs and they're positive, then I would never complete a loop.
Okay, in that case, then why should you actually minimize some ofCijs? Meaning you can try to find a graph with some connectivity property whose cost is the minimum, and not necessarily a tree always, right?
So why we use the Steiner tree? Because we thought that that was a simple thing to do to try to identify some additional interesting nodes that would come out in a minimizing solution. You can try to model it with some other combinatorial optimization problem and go through and see. I mean, this happened to be a simple way to do it. The thing about these biology problems is I'm not saying that the Steiner nodes are the underlying biology. I'm saying, or that the Steiner tree is the underlying biology. I'm saying the Steiner tree is a way of finding possible proteins that are important in a particular pathway, but that weren't previously identified because they weren't underexpressed or overexpressed. Maybe at their background value, that protein is a catalyst for something else, just at its background value. So nobody would have identified it. But I'm just trying to find a method that will identify particular, possibly interesting proteins. There's another combinatorial optimization problem that does it. That would also be fine. And there probably is out there.
Thank you.
Can feedback loops be captured by these processes? Because trees obviously don't have cycles, but after superimposition, can the feedback loops be captured?
So, yeah, and we expect the interactome to have feedback loops. Okay, so that is getting captured when we superimpose these things. So yes, it is being captured. Now, there's a big question about the temporal sequence, which was asked. And so I don't actually know that this is a feedback loop because maybe I'm imposing things that are in the wrong temporal, superimposing things that are in the wrong temporal order. So when I have accurate enough data to begin to get the temporal data in there, it may be that some things that look like a loop are actually not a loop for temporal reasons.
So these biological networks you showed, like these examples, so what is the degree distribution? What is the degree distribution there?
These most of the biological networks that people have found look like, I mean, most, you know, are around 1 over k squared. I mean, there are some, so in the competition-induced preferential attachment paper that we did, we actually gathered lots and lots of data from different domains, and we found that the biological data clustered around certain numbers. It was about like 1 over k, but it wasn't at all uniform.
And also these degrees of separation between like two nodes, average degrees of separation?
I don't know what those were. I'm not sure
but it's not
what those
6.
were. I don't know, and there's nothing to say that we're getting the whole network. So we may only have a subset of the network. So I wouldn't believe that either of these numbers are necessarily an accurate reflection of the true network.
Okay, so if there are no other questions, let's thank Jennifer again for a very nice talk. Thank you all for coming. There is some tea outside on the other side.